This protocol facilitates the development of a machine learning algorithm for predicting the growth of bone metastases in an experimental model at the stage of early organ colonization.
The main advantage of this technique is that it combines several imaging parameters into a machine learning algorithm that significantly outperforms the predictive ability of each individual parameter.
Though this protocol aims for the early diagnosis of metastases in bone, it can be adapted to different organs or areas of multimodal and multiparametric imaging research.
"10 days post-surgery and injection, in a DICOM viewer with a DCE plugin, click Import to load the DCE sequence in 4D mode and select the DICOM folder containing the MR images. Place a circular 1.5 square millimeter two-dimensional region of interest in the proximal tibial shaft bone marrow of the right hind leg within the 4th or 5th MR image and select Relative Enhancement in the plot type. Enter the baseline range from time points 1 to 5 into the respective fields and export the analysis as a .txt file named dceraw.txt. Open RStudio and load the provided DCE script.r file. Select Code, Run Region, and Run All to run the entire script and copy the output to the provided imaging features template file. In the DICOM viewer, place a second region of interest within the back muscle of the animal and repeat the DCE measurements as demonstrated.